Hey students, how are you doing? I am Nishtha Gupta from IISc Bangalore. I have been working with askIITians as a biology faculty since past 4 years. Today we are going to take the topic muscle contraction. This topic will be divided into two videos, and the first video we will take what is a muscle fiber, the parts of a muscle fiber, and we will discuss about the muscle proteins. In our second video, we will see the complete process of muscle contraction, which is known as the cross-bridge cycle or the sliding filament theory. So let's begin with the first part. Muscle contraction. We can see here a skeletal muscle fiber is shown in this diagram. Sarcoplasm is here, sarcolemma is here, and nuclei are present in the muscle fiber. Also, various myofibrils are present within a muscle fiber. Let's have a look at the zoomed view of this part. This myofibril is the main site of muscle contraction. Thin filaments and thick filaments are present in a myofibril. These thin and thick filaments in turn leads to muscle contraction and relaxation. How this will happen, we will discuss in our next slides. Before leading into the theory of muscle contraction, let's see what are the muscle proteins. A muscle fiber is composed of 20% protein, 75% water, and the remaining 5% in a muscle fiber is composed of inorganic and organic matters. We are having muscle proteins myosin, actin, tropomyosin and troponin. Let's see what is myosin. Myosin is the compound from which thick filaments are made. So, thick filaments are made up of myosin molecules. It contains two identical heads. Okay, myosin contains two heads and both are identical with pairs of helical strands which form the tail. We will see in a diagram how this myosin structure is formed. The upper surface of each globular head contains actin binding site and ATP binding site. So, the upper head of myosin, the two identical heads are here, and the upper surface of each globular head, it contains actin binding site and ATP binding site. With these sites begins the journey of muscle contraction. Actin forms the thin filament. It resembles two strings of beads. So, two strings of beads will be there which are twisted into a double helix, and actin shows high affinity for calcium ions. So, when they are having high affinity of calcium ions, they will be helping in muscle contraction. Tropomyosin is also a type of protein which is two-stranded alpha helical rod. So, it is two-stranded alpha helical rod, and it is located where it is located in the groove between the two helical strands of actin. So, two helical strands of actin, between that groove tropomyosin will be found. Troponin is the globular protein. It consists of three polypeptide chains. What are these three polypeptide chains? The calcium binding subunit, the inhibitory subunit and the tropomyosin binding subunit. Let's have a look at the diagram so that it becomes clear. Thick filament. First of all, have a look at the thick filament. This is myosin. Two heads, two identical heads are there. In each myosin you can see two identical heads are there, and this is a coil string. This is the tail. This is known as the myosin tail, and these two identical heads are the myosin head, and these myosins form the thick filament. In thin filament we can see beads are there. two beaded strings are present, and these two beaded strings are of actin molecules, and they are forming the thin filament. And you can see troponin is here. We have seen in the last point, troponin is in between the grooves of the actin. Tropomyosin is this alpha helical strands. You can see alpha helical strands. These are tropomyosin. And actin binding site and ATP binding site. These sites are present in myosin head, so that they can bind to the actin and they can bind ATP in order to let the process of muscle contraction happen. Okay, so this was all about muscle contraction part 1. We have discussed a muscle fiber and we have seen what are the muscle proteins. In our next lecture, we will see the complete mechanism of muscle contraction known as the sliding filament theory.